Thank you very much, Dr. Hiền Phạm and your team. Powered by deep learning, modern AI has enjoyed great successes in cognitive domains such as computer vision and languages where humans can naturally perform well. But for the field that traditionally require deep expertise such as physics, chemistry, and biomedicine, how AI can contribute to that? Dr. Truyen Tran, Associate Professor of Artificial Intelligence at Deakin University, will give us an overview on modern AI for drug discovery, how AI is speeding up and lowering the cost of this field. Please welcome on stage Dr. Truyen Tran.
Thanks so much for the nice introductions. I have to blame Tang for two things. One is that he placed me at the end of the day where everyone is tired. I asked for the afternoon session, not the last one, but I consider the lucky one. All right. Another one is I blame his employers for making me working on this topic because they saw it all. They've done all computer vision and NLPs. Then I ask myself, what's left for me, right? So naturally, it's left for me is biology, right? So then here we go as a drug discovery. It's funny thing because when I search the keyword drugs, it's just to find some supporting documents, nice image for my slide, and they're all about drug dealers. And so this one is not about that, right? Don't be too excited. So my name is Truyen Tran, and I got introduced. I'm from Deakin University in Australia. For those who don't know where it is, it's near Melbourne. It's a city in Victoria, in Australia. So, just a few months ago, there was a big breaking news. I think it's as big as BERT, right, in NLPs, or maybe comparable to AlexNet in computer vision. So it was the moment that, for the first time, AI actually suggested a drug, and it was made in the lab, and it was tested on the mice, and the result was good, right? So, and the total period of time for doing so before the testing was only 46 days. It's a record terms because normally it takes like years to do so, right? So it's the first moment that we really have something that created from AI that actually works on the mice, not human. Yes, don't be too excited. It takes many years to test on human. That's how thing works. Right. So, so my talk today, if I have enough time, I've been told that I don't have enough time. So I will introduce a little bit about the field of drug discoveries and how to proceed from there, from the AI point of view, because I'm not a biologist, I'm not a chemist, so I can't talk from that point of view. So from my point of view, the things that AI people would do, first you will learn implementations, like you learn implementation in any fields, and you want to predict something, you want to and generate something. So that's pretty much AI view of the world. So for those who don't know, drug is a small molecules that binds to a biological target. Could be most of the time the proteins, and could be something else, but protein is a common one, and that modifies the function of that target so that it has the intended effect, right? So in that definition, tea is or coffee is a drug because it modifies your mental effect, the mental powers, and have intended effect that probably you know it. And protein is a large biomolecule, so it diverse way, right? So one is the more small molecules, another the big molecules. So the main different thing is that this is a change of the amino acids. Right? The drug is normally thought of as a graph, and protein is pretty much in the broad form. It's a string, right? You can think of it as a string of text. And here it is. So drug is small molecule. Protein is the big folding things, like ribbon folding things. And when they work together, the drugs kind of like dock into the protein and have some effect on the protein itself. So a drug discovery is a process of finding a drug with the intended effect. So pretty much it's about that. But it's very complex in the sense that you have to understand biologies, chemistries, and almost everything else to make the drug work. Right? So that's why it's expensive and it's very important. There's the concept of drug likeness. So to be able to quantify as a drug, the molecule have to satisfy certain properties. That's not all, but at least some property have to be there. Like you can put it in water and it works, and it goes through the body and it goes to bloodstreams. So it have to be small enough. It have to have certain properties as well. So biologists would know those properties well. And here's the traditional highlight of discovering drugs. First, it takes about 10 years or more to start from the concept to the final drugs that being approved on the market, and it costs about $1 billion or more, right? And probably it costs a lot of money in the process. And it's very expensive because you have to scan through like 1 million molecules to get about 1,000 of possible molecules, and then have from the 1,000, it probably that one of them would make possible drugs, right? It's very, very expensive process. And then come to the AI-driven process, which is very, very new. It's been there for a long time, but only speed up like I think about 3 years ago, right? Then it's quite different. Right? Here is a process that proposed by In Silico, is one of the leading AI pharmacy company startup right now. They propose like using AI throughout all the steps of making the drugs. And in our talk, we are focusing mostly on the middle part and how to identify and generate a possible molecule that has the intended effect. This is quite complex, but interesting, very interesting. Right. So in this field, we try to answer 3 basic questions. One is that if you have to give me molecules, and is that the drug, right? Or is it just random molecules? Right? So we have to answer all kind of other questions like, does it look like a drug? Does it have the target to bind to? Does it work on the target? What are the properties of the drug? Like when it goes to the body, what does it affect? Like, does it make you happy or sad, or how does it like go with food, go with waters? How does the body accept or reject it? A lot of properties that we have to answer before we can quantify something that worth considering, right? Another question which is much more difficult, that given a target, which is a certain protein that we want to modify, what is a molecule that can affect that target, right? It's a very, very difficult question because researchers, they identify the disease, and from disease they identify certain bio-target, like certain protein to modify, but then no one knows how to do it, right? No one knows how to make a drug yet, so it takes many, many years to do so, right? So if the list of molecules given, so we just rank them up and pick the best one. But most of the time it's not given, right? So we have to suggest a molecule which in the form of chemical formula with the hope that we can make in the lab, right? It's no guarantee because it's not easy. So the question is that given a molecule, if we know one, then how can we make it, right? So it's another question that AI also contribute to. It's called synthetic structurabilities, and for those who keen on AI reasoning and planning, it's pretty much planning problems. Right? You start from the goal you want to achieve, and you work back. What are the steps that from the very basic molecules work up through the reaction chains up to the molecules that you want to. Right? So it's not. It is a planning problem, but it's a very interesting one. Right. So like anyone who work in AI these days, you always start with representation. Right? You don't start with raw data anymore. Right? So given the molecules, you want to put the molecule in the form that computer can process it, right? So the first thing we do is, of course, put it in a vector, right? Embedding all kinds of tricks that you do. Chemists, they've been there for a long, long time. They actually have a very nice way to do so. They call fingerprints, right? So fingerprint is a way that convert the graph into a vectors, a discrete vector, basically. That's because it's very easy to deal with vectors, right? We have all kind of mathematical tools to deal with vector. You can transform it, you can multiply, you can delete, you can add random noise to it. And here is one of the techniques that's more recent. Actually, it's not a product from biochemistry, it's a product from machine learning. It's applying the convolutional nets to the graph molecules, and then in process compute the vectors. But it was a very early days. And then once a vector, you can do a lot of things, like for those doing deep learning, you know that you can do autoencoders, you can do the GAN, you can do all sort of other things. And this one, in one example, using the adversarial autoencoders for generalizing the new fingerprints, right? They work directly on the fingerprint itself, not the graph. So that's one example. Then another way, even more popular way to do so is that you either give me the molecules, I try to represent it as string so that a computer can read it. That's just like normal sentence, right? So one of the most popular one is called SMILE. It's a way to convert a graph into string. You see the molecule here. You can and here the one way to put it into string. Then multiple ways, not one way. But this one, computer can read and understand it. There's certain grammar rules and to make that this kind of representation. It can go sentence if you like, because it has a certain grammar. Because there's sequence, so as a deep learning person, you can think, well, okay, I can put in sequence to sequence model with attention or other tricks. So transformers and many other tricks, right? Even memory networks, even reinforcement learning, that all tricks that you can think of because that's sequence, right? But the problem with that, the string to graph is not unique, right? And the graph to string is not unique either, right? So you have the trouble. And if you generate some strings, a lot of them are invalid, right? No way you can convert back to a graph with some string, right? Because that space of string is kind of like very, very unrestricted, whereas the space of graph is much more restricted. And the precise information, 3D information, is lost. And 3D information is very important to compute a lot of dynamic property of molecules, right? And the other trouble with that, if the graph, you have a certain short range between the two atoms, but then when you put in strings, when you put in strings, certain atoms are quite distant in the strings, whereas in the original model, it's close, right? So the better way is, of course, it's using the graph by itself, right? You don't need to do anything, just represent the molecule as a graph as it is, right? Troubles, again, right? Because graph doesn't have a fixed size, right? It doesn't have fixed orders, right? It can permute and it's still the same graph. It don't have very good model of generating a graph, because it can have a good model of autographic model of generating a sequence, but it's not easy to model a distribution of a graph because of the all trouble that we know, that in no regular structures, no fixed size, and no easy way to fix the permutation invariant problems. Okay. And yeah, okay. There are many other requirements as well, but we're not talking about this for now. Right. So here's one of the things that we work on for the last 2 years. It was inspired by the memory networks, a classical one, like about 4 years ago, 5 years ago. But we tried to make it to work on the graph structure data, and quite generic. So it could process a graph in a nice way. So here's one example. So not example is here's how it look when you run it, unrolls and run it with your deep learning frameworks, and it become a computational graph. So it receives the drug graph as input, and have the controller to control the message passing between the molecules. So the atom to mimic the interaction, the bonding, chemical bonding between the atoms. And at the end of the day, it collects the output, and that is either prediction or something else, or generation or something else. Quite neat frameworks and to do many things. All right, so that the molecules, which is the drug itself. But then another question is that big question is how to represent a protein itself. So for those who are not in the biologies, protein is a strings of amino acid and represented at like 20 different characters. We have only 20 amino acids in the protein, and you have a string of characters. The vocab is about 20. And the length could vary from, I think, few hundred to few thousands. But then the problem with the 1D representation is that it doesn't look like a real protein. The protein have the 3D structures, right? But the problem with 3D structure is very hard to know. No one have very good model yet of how to convert a 1D string into 3D. So the latest and winning method so far has been default from DeepMind. but then it's still quite inaccurate compared to what we really want to have. And the practice that we have, and very few proteins this day available, those are being characterized by empirical methods in 3D structure. But we know that we on 1D structure very well. But then the good thing about 1D structure that you can actually employ a lot of NLP's techniques like embeddings, doc2vec, close, sequence2vec, or other MO, the BERT, or other advances you can think of borrowing from the NLP field. So actually it's very, very popular research this day how to use NLP technique to represent the protein in a structured way that is useful for the future work. So I'm moving on to the next part. So the question is that if I have a protein and then I have a molecules, and what are they doing together, right? So we expect that they interact, right, in certain way, but who knows, right? It's not easy, right? So the job could normally call the drug target binding or drug target affinity prediction or many other things, but from a machining point of view, we can translate the problem into question answering, right? So Tang here would be an expert in that, so you can tell easily, right? So, all right, you have the questions and you have the text, and by nature they can put together, right? So here's one of the model that actually one of colleagues of mine cook up, and it's quite simple. You use a graph convolutional net to process a graph, and you use CNN to process the string, put together and predict, right? So quite simple, quite intuitive, but it works quite well, right? So I put it here is that you can think of as question answering. You have the context or text, or you have the queries or questions. I have the answer, right? The answer here is pretty much the scoring, the numerical scoring of how good or how strong the interaction is. We also make use of our own structure, our own models to make things more flexible, because we can put as many queries as you want. You can put as many graphs as you want. The model actually can be extended into multiple graphs. And then you can train jointly. We can do many things. So so the result was here. Some result was here, and it showed to outperform all the known techniques so far in predicting whether a drugs interact with certain targets. So that is called RDMN. Is one of the techniques that we developed so far. So another way to think about is drug is still binding is still a QA problems. So here's another techniques under development. So we think of the protein as a big structure which have many parts interacting with each other, and also at the same time interacting with the drugs. So here's a technique that the model that we are under development. The results so far is quite encouraging, but we're not finalizing yet. So the idea is quite simple. We have a unit called randomized unit, which transform one array of objects to another array of objects, condition on something, right? Condition on question or queries or any other things, right? And the protein being here that considered as a hierarchy of the random power set, which is the concept that we cook up. And by doing so, we're bypassing two things. One is that you have to estimate the protein structure, which is we don't know how to, so we just try to bypass the problem. Another one is that we don't know where the binding happened, because the drug is small and protein is big. So when the drug comes to protein, it's only bind to one tiny bit of the protein. We don't know, right? So we have to make assumption that something may happen, and we try to aggregate all the possible effects. All right, so moving to the next part, having predicted the target, the binding effects. The next question is that how can you make use of the known drugs for the new purpose, right? We know that because it's very expensive to make a drugs, right? $1 billion business. It takes many years, right? So what if you use a known drug for something else? So that's called repurposing. And actually very popular this day. A lot of drugs being developed for one thing being having the nice effect to another, then so you can make use of that for free, right? So again, we made use of the techniques that we showed before. Try to predict single drug affects on many things that possibly we can think of. So that those cancers and the fungus things. So we show that training jointly together is more accurate than training one output at a time. Right? So two things. One is that one drug can actually work in many things. Another one, the thing is that training jointly can give better results. It's quite a well-known effects of multitask learning and similar things in machine learning. All right, another one is another model which was published early this year. It's called the GAML. It stands for the graph attention or something similar for the multi-label learning. So I'm not going to detail here. The idea that you can expand the number of possible outputs as many as possible at a very minimum cost. And the effect is quite similar. We can have good result by predicting many outputs at the same time. Like let's say here, 9 cancers at the same time, or binding to 50 protein at the same time. Right? You do at the same time, actually more accurate than doing one at a time. And that's very nice result. All right, so another issue that in drug development, you have to be worried about the unintended effect of interacting between drugs, right? So if the good doctors ask you what, before giving the drugs, normally they ask what are the other drugs that you are taking, right? Because they actually interact. Many of them interact in a bad way, right? Unintended way. Some are interact in a good way, some are not, right? So we need to know in advance that what are drugs are interacting or can predict the property in advance. So in doing so, it creates another problem. We have multiple graphs, which each graph is one drug, and you ask the question, what are the pair of interacting? Or if it's not the pair, then if it's with them like triple, right, interacting at the same times. And it's going to interesting problems. And in doing so, we actually, it's not showing here for some reasons. But then we extended the RDMN framework to incorporate multiple graph, which is helping us to handle multiple molecules at the same time. And we apply it to predict the chemical interactions. And the result is quite interesting because the model is flexible to incorporate many other features as we know, the contextual information and features we know, and other prior information we know about the environment or the structure of the graph. And the result was very interesting from the standard data set compared to other techniques known just this year before. Right, so coming to the last point. This is the most important part and most difficult one, and there's a lot of activity going on right now, and success or break of AI pretty much depend on this, because the prediction is easy, but generation is not, right? So one way to think as an AI person, you can think of drug design as structure machine learning translations or conditional generation, if you like. The only thing is that the drug itself is not a sequence, right? It's a graph. So the generative model of graph itself is very, very open and important topics in machine learning these days, right? Well, it poses a question of what kind of representation you use, right? If it's string, then yes, it's very ready. Technology is quite ready. But if it's graph, then it's not easy, right? Because we have the models in node interaction at the same times. You have the model many other things at the same times. If it's sequence, then you can actually treat the graph as a sequence in a certain way by stacking subgraph together in a certain sequence. You still recover the original graph. And the good thing about the sequential treatment that you can use reinforcement learning techniques, right? It's kind of like a sexy technique these days. Anyone doing AI dream of applying reinforcement learning to their problems. Some works, some doesn't, but that's the reality of the day. Right, so another problem is that when you have some structure that you hope that become a drug, then you have to optimize it because it doesn't come easy, right? Generative model sometimes generate something very weird, right? You have to optimize it for the purposes you want. So the problem is called molecular optimization, and once again, you can think of as a machine translation, right? It's very interesting. So here's an examples. You have a original graph. You can convert into junction tree. For those who don't know, junction tree is a super tree where each node is a subset of the nodes, and then you can actually using encoder and decoder architecture for tree, not for sequence. Okay, this is known now, so don't think of inventing it again. And then you can actually recode it from the junction tree, recode back to the graph. Right? So the trouble here is that the molecular space is very big, right? Probably bigger than human languages. So the modern, one of the latest technique, I think ICR this year is, I find from MIT's, they treat the molecular optimization as graph and graph translations, right? So people know, probably in machine translation, you know the sequence to sequence mapping is translation problem, but now we have a graph to graph mapping as also translation problem, right? And they have quite interesting results. So I'm not going into detail of that, but the next one here that we can translate the problem into some other form that we know before and apply the optional techniques or probably with some cool, change to the Docker architectures, but the principle is the same, right? And then, same AI technique can be applied to both NLP and drug design. Right, all right, so one of the very busy space is using variational autoencoder for drug modeling. This is exciting just because the dream of AI is to explore the entire space. Right? Anyone working in AI is they want to explore the entire space of something, right? But it's not possible. Not that easy. So for those who don't know VietAI, here's very simple graphical architecture. I'm not going to detail that. But this is actually one of the big moments that people prove that instead of dealing with drug directly, you actually can dealing with like a continuous space, small continuous space, something like a 10-dimensional. Whereas you know that the space of drug is very, very huge. Whereas 10-dimensional is quite easy to deal with this day with the modern optimizers, right? So what they do with that, they convert the drug into string smile using INN encoder, put in the vectors, and then put it back, right? So, and then it can actually optimize on this little vector space so that it can move along the line of discovery, right? And here's some example what they did. The optimization was done through Bayesian optimization or another technique, but you can use any technique you like, right? The effect is almost the same. The lesson here that even the drug space is discrete and very big, you can actually convert it into much smaller continuous space and using the whatever technique you like. Because you know it, right? So, and it still works. It's very, very interesting. Another lesson so far with that, instead of dealing directly with drug, you can convert it to tree, as I mentioned in tree before, because tree is much easier to deal with, much nicer. You can use, because you have a wild form structure, and you can have forward, backward kind of operations. Very nice. And the last one is that you can actually use reinforcement for the graph if you treat the graph as a sequence of smaller subgraphs, right? So it looked like thing can be done, and here was actually the result from the NIPS NeurIPS last year, obtaining the state of the art 1 year ago. The field is really going really quite fast, like comparable to QA field. So every few months you have some state of the art, and probably a few weeks now you have the state of the art. So this is already old news, like 1 year old news. But it's one of the significant ideas because it's a demonstration that you actually use reinforcement on the graph domain, and it works quite well. Right. All right. So the last one, probably not really the last one, but quite close. The question is that you have the drugs, you have the molecules, but it's not yet a drug, right? How can you make it? Right? You can make it in a computer, but how do you make it in the lab? Not easy, right? So here's one idea that actually predict not the drug itself, but the reactions. They predict the components so that it can react to make another molecule, bigger molecules, right? So there was a papers just on the archive, I think, a few weeks ago. But luckily, there many work on reaction prediction itself. We can actually predict if the two molecules interact, react in a certain way. And here's one of the techniques that we develop and published middle of the year. So it's called the ZTPN. It stands for the graph transformation policy networks. So to predict that if the two molecules go together in given some a reactant or a catalyst, what are the product of reaction, or if they interact at all, right? So the accuracy was good. So we can plug this model into this framework and ask a question that if I do the drug design, then can I make it in the lab? So in the entire loop of discovery itself. So for those who are really keen on playing this field, here is one of the most comprehensive frameworks, so that you can play with most of the known models so far, with the data set that people collected, and with the matrix that people cook up in the field for the last few decades, right? It's called Moses, and that's it for me. Thanks very much.